This technique allows for not only localization, but also quantitation of candidate mRNAs in individual oocytes and embryos. Compared with other assays, including PCR, completion of the technique results in reproducible data with low variation. Demonstrating the procedure will be Kelsey Timme, a graduate student in my laboratory.
Begin this procedure with the preparation of the required material and female mice at 5 to 8 weeks of age, as described in the text protocol. Clean the mouse using 70% ethanol. Expose the abdominal cavity and visualize the female reproductive tract. Hold the ovary with forceps and remove the uterine ligaments and excess adipose tissue from around the ovary. Cut the oviduct from the uterus. Then, place the ovary-oviduct pair in warm holding medium in a 35 mm dish. Remove the ovary and any surrounding adipose tissue. Tear the swollen ampulla of the oviduct using a 1/2-inch 27-gauge needle. Push the oviduct at the site of the tear, and the cumulus cell oocyte complexes will be expelled. Use a mouth pipette to transfer the ovulated oocytes to the 100 microliter drop containing holding medium with hyaluronidase. To dislodge cumulus cells, pipette the metaphase II oocyte cumulus cell complexes up and down in the hyaluronidase-containing holding medium with the mouth pipette. Once they are devoid of cumulus cells, use the mouth pipette to transfer each oocyte to a wash drop containing only holding medium. Repeat this.